The JOIDES Resolution is a 500-foot drill ship, and we use that to take cores of the seafloor. And so we lower a drill string, and we drill into the seafloor and take cores up through the middle of that drill string. My name is Adam Klaus, and I'm an expedition project manager. The place where we're drilling is the interface between the inside of our planet, the stuff below the seafloor, and the oceans and the atmosphere. So there's a lot of challenges with implementing this type of expedition. Will we have trouble? I'm sure we will. These are very initial experiments to try to get the baseline measurements of what lives below the seafloor, what lives in the sediments, what lives in the oceanic crust. It's something that we don't know anything about, yet we know that it takes up a third of the biomass on Earth. All the holes drilled in the world's oceans represent the vast archive of Earth's history. This goes on and on and on. Atlantic, Indian, Pacific, as well as Mediterranean oceans. If we take out and look at one of the cores, sediments have been laid down layer by layer, and the scientists can come back and determine things about the Earth's history. It's a very important resource for the science community.
My name's Julie Huber. I work at the MBL as a scientist, so I run a lab that studies deep sea microbiology. The Origin of Life community is interested in finding microbes that might contain genes or traits of the earliest organisms on Earth. The oceanic crust has been around since our planet pretty much formed. There is virtually zero data, about who's living in those rocks, who's living in the fluids that are covering the rocks. Given that 70% of our planet is covered in these rocks under the ocean, it's a big gaping hole. The water right behind us at some point in time has passed through the ocean crust. And so it's just this huge conveyor belt's the best analogy I can think, just where every parcel of water is moving through this large mass of rock. You think of a jar of marbles, so there's a lot of space, and so the water's flowing through that space. And I'm just interested in who is there and where are they coming from. But we don't really know because we've never looked in that type of detail. And that's what we'll be able to do with this expedition.
My name is Katrina Edwards, and I'm a professor of biology at the University of Southern California. Most of the species that we've observed so far in the subsurface are completely novel. It doesn't look like anything that we know in the surface world. So if you can imagine that Australia was somehow completely cut off from human exploration, we'd never discovered it. Some of it may have similar functions to the surface world, but we don't know because we haven't explored it as of yet.
So my name is Peter Girgis. I'm a microbial physiologist. Humankind has always had an interest in understanding the world in which we live. And to do that, we have to leave our homes and go places and see the world around us. We try to figure out how things work and how well they perform. So if you think of all the places where animals and plants and microbes live, 80% of all of that biosphere is actually deep ocean, and we really don't know a lot about it. Without microbes, especially like microscopic algae and the like, we would not have the oxygenated atmosphere that we have today. So at the end of the day, one can't have a conversation about climate change without seriously considering the role that the ocean plays in it.
My name is Heath Mills. I'm assistant professor here at Texas A&M. We're going beneath the surface of the ocean and seeing life that no one has ever seen before. Well, if there's life on this planet beneath the surface of the ocean, what does it mean to be alive elsewhere? These are fundamental questions NASA is addressing with, is there life on other planets? We've found a way into space. We've marched our way into and around the solar system. What would happen if we stopped? What questions would we lose ability to answer if we no longer sent probes and sent man into space? The same thing happens here. We have so far only scratched the surface.
The hypothesis is that approximately 1/3 of Earth's biomass carbon could be buried in the subsurface biosphere in the marine realm. There's the realm that we have essentially no data at all. The overall mission to North Pond is to go to sample and start experiments at a site in the middle of the Atlantic Ocean. It's actually a site that's been drilled, it's been examined, it's been studied by scientists for the past 30 years.
This is leg 28 of the deep sea drilling project.
We get to finally test basic hypotheses about the role that microbes play in transforming the ocean crust into something else. And we've made observations as a scientific community for decades about that, but nobody's been able to actually go out and address the hypothesis using explicit experiments, and we get the opportunity to do that. That's what I think is cool.
Oceanic crust, we know, is very challenging. If anybody has ever been to Hawaii or seen the Big Island where volcanoes are happening, the oceanic crust looks something like that. And so we're drilling into this broken up rubble, which is not the easiest kind of formation to drill in. And so that creates a lot of drilling challenges. Putting observatories into the oceanic crust is probably the most challenging environment we can do that in.
We're going to be going down about 500 meters into the igneous ocean crust, which is about 4.5 kilometers below the surface of the ocean. So 2.5 miles.
My name is Tom Pettigrew, and I guess I'm called the Cork Engineer, if you will. A hole is drilled in the seafloor, and then the cork seals the top of that borehole so that the scientists can sample fluids from downhole, monitor pressure changes, temperature changes. The scientists are always wanting to go further with their experiments, and they can drive me batty at times with some of the things they want to do, but looking forward to getting back out there and getting these things in the ground.
The scientists want this really bad. They've been working at this for years. Kyra Becker is a crustal hydrogeologist. He studies fluid flowing through the rocks. Kyra has a vast amount of experience working with all the people on the ship. He's sailed more than any other scientist. I think he's on his 19th expedition. Katrina Edwards is one of the 2 chief scientists for this expedition. She is a fairly unique scientist and person in her field. She's very focused. She's very interested in not wasting time to get it done. Katrina and the other co-chief scientist is Wolfgang. They're like telepathic. They've been working together so long.
Wolfgang's my partner in crime. He's a professor, a petrologist, and a geochemist from the University of Bremen in Germany. Wolfgang and I go way back. We actually originally conceived of this project together. He's patient, he's funny, he's kind, and he makes me coffee every single day.
Heath Mills is a microbiologist at Texas A&M University. He works mainly with deep-sea sediments, which is something very difficult to do. And it's also something very important to do because you learn something about what microorganisms actually do and if they were alive. So good luck to Heath for getting something out of the samples.
Beth is so much fun to work with. Organization doesn't begin to describe Beth. She is so organized with everything she does. She really is able to do her science, to help you do your science, and put it in perspective to a way that in the end you're like, oh, that's what I was trying to do. And she really pulls off very, very well the lead by example. She'll be demanding of you. She will make sure that you get your stuff done. She's going to be working there shoulder to shoulder with you, right beside you the whole way through.
Jeff Weed is an interesting human. They only make one of most humans, but they definitely only made one of Jeff. Jeff is a kid in a candy shop when it comes to doing science. He's a geochemist. Jeff's been responsible for almost all of the Osmo sampler, the water sampling apparatus, which is not a small feat at all.
I knew Sam when he started washing bottles for me when he was an undergraduate, and then I was his advisor, so I've been interacting with him for many years. Sam's an interesting guy. He has evolved over the years, let's put it that way. He's got his own little experiment now. He's got a fast flow Osmo sampler that he's going to be putting out. So this is his first experiment where, you know, he's the lead and he's the guy who thought of it and he's the guy who's pushing it, and so it's fun to see that.
The scientists on this expedition come from all over the world.
Nice to meet you. Welcome. Thank you. Sam, how you doing? Good to see you.
We have scientists from Japan, from Europe, the United States, Australia, China, Korea, and India. We're going to spend 2 full months at sea.
I have no idea what it's like to spend 2 months at sea with people I don't know or I've just met. This is my first time. I've been out to sea many times, but they've only been short trips.
It's hard to predict how any of these type of expeditions will go.
Your responsibility pretty much starts years before the expedition starts. You're full with anticipation. You look forward to get going.
I'm extremely excited about this mission. I'm pretty nervous also about being away for 2 months and my capacity to stay self-entertained in a captive environment.
All of this was building up to here's my first cruise. I mean, this is my chance to put my money where my mouth is.
When we're out on the ship, we can't order any more parts, so it's kind of do or die time at that point.
Right now, there's still a lot of anticipation. There's so many things that could still go wrong. We haven't even gotten to the site yet. We could have trouble drilling. We could have trouble installing the cork. We could have trouble getting the instrument string down. So every one of those things, you're kind of keeping your breath held a little bit, like, is it gonna work?
Now we're going to actually really get started. I'd like to welcome you all to Expedition 336. It's been a culmination of a lot of work from a lot of people, not so much me, but people like Katrina who have been pushing this proposal for so long. And the very first thing we're going to do is go to hole 395, and that's one of, Kyra, of course, has been working in this area for many decades. And we're going to pull that old cork out, and it has a thermistor string. Kyra wants to get his thermistors back, I think, and I suspect microbiologists want to take pieces of the thermistor string or samples off that. The next thing we're going to do is a logging operation before the hole is any more disturbed to image the borehole walls to look for bacteria. The very next thing we're going to do is we're going to install a new cork. So we want to make sure that we pull this cork well and install the new cork really well. And so this is all you guys. So Bill has some things he wanted to talk about, and I know that Kyra and Jeff at least have some questions and things, and that's why we have all the operations people and Tom and Bubba here.
We are running fiberglass casing for the first time in the program's history, at least anybody will claim.
We don't know what this fiberglass is going to do. And my greatest fear is we break a connection, and then all those umbilicals are going to spin off with it.
So, and also, there's a big assumption here that this cork is not latched in. So, from all records, I think they had tried 5 times. I think this cork is not latched in. So, that's our assumption right now.
On the whole, you guys kept tagging around 600.
Yeah.
And that's been stable for 20 years. So.
It's been 14 years since the last time.
Yeah.
Right.
The only thing I can think that could be, change the game plan is if we learn something new at WT.
There's also, if for some reason a large part of the bottom of the hole is unacceptable,
Yeah.
and so I guess what
What do we do?
What do we do then? I mean, you know, is there?
There's already 100 meters or so I mean, the deepest Cork part is 100 meters above the hole bottom or something, isn't it? So there's already 100 meters slot there. Yeah.
If anybody has any questions or concerns, let Bill know. The sooner the better.
This is totally extraneous, but we want to decorate the corks like we did last year.
Man, I'm not the guy to ask them.
Yeah, no, I'll handle organizing it, but I need to know when.
Certainly, you guys need to make sure that you're comfortable with how she's doing it and where she's doing it. So, okay.
Yeah.
Thanks, everybody. That was rather short for us, considering all we got going on.
My name is Samuel Holm, and I'm a researcher from Moss Landing Marine Labs. The original drilling in North Pond really had nothing to do with the deep biosphere. They just needed some mud to hold their drill bit stable while they drilled into the ocean crust.
After they left the hole, it immediately started to suck seawater into that hole at the rate of about 20 liters per day. And it did so for the next 20 years. So we learned water is flowing into the ocean crust in this part of the world.
My name is Kyra Becker. I'm a professor of marine geology and geophysics at the University of Miami. We're setting up a hydrologic shunt, allowing the ocean bottom water to go straight into the formation. By putting a plug in the top of the hole, we could actually understand what was really happening in situ, in place, in the formation, before you drilled the hole. So this has been in the ground for 14 years. I was actually the chief scientist when we installed it in the first place. Now with the new emphasis on the microbiology, we had to install a new model.
Pretty amazing, actually. You're trying to find a target that's 12 feet in diameter from a moving ship. Most of that is done blind.
Attention on board, start lowering thrusters. Start lowering thrusters.
With a drill string that's hanging below that ship, which is affected by currents and movements of the ship, we're talking about a 1 meter diameter tool going over top of that wellhead to latch it. And they're able to maneuver the ship with that degree of precision to move that pipe to where the driller can lower it over top of that wellhead. They make it look easy, but it's quite an accomplishment to do that.
Some power. Right now, everything's looking great. Pressures are acting like they should.
Got my fingers and toes all crossed.
There you go.
Bubba and Wayne and the driller will all be conferring.
We'll get down to seafloor so we can see it, and then we'll start a search pattern to look for the cork. Yeah.
If it's latched and he picks up, then he should pick up the cork. If it's not latched, then he'll come off of it. Well, we've all been cautiously optimistic that everything's going to just come out like it's supposed to. Chances are it's going to be stuck a little bit. I mean, I'm not sure we'll get the sinker bars out. Depends on if the hole's collapsed around the lower part of that. With our positioning capabilities these days, if those sites are located accurately, I mean, we're real close.
Well, it's not
45.
perfectly centered.
No. All right, what do you want?
Ready floor. Hey, this is Charlie. Hey, this is Jake with DP. Hey, you guys ready to go?
Yeah, I'm full 2.5 meters in on the knobby right now.
We just want to let you know it looks like that cone is right.
Yeah, it's right. Looks like it's right to the left.
Okay.
I mean, we can probably move a little away from it if you want to. Look.
You want to have them just move over a little bit?
Yeah, just in case we do bump it or something up.
I think it'd be a good idea.
Okay, I'm gonna go ahead and move it here just a little bit.
Oh man, that's real close right there, huh?
Gonna try to turn to the left just a little bit. I've got a little bit of weight on it.
You just gotta get us.
Camera's gotta always be on the wrong side of it, doesn't it?
We're nipping at the edges.
All right, Phil, I'm gonna start coming down real easy, huh?
All right. Now look what you done, Charlie. I can't see it.
That wasn't bad. I don't know. That was pretty damn good. Yeah. Okay. Let's go see if we can get the thermistor.
Okay, the thermistor string is basically an electrical cable that has 10 thermistors that are temperature-sensitive sensors. I had made the sensor string. They should be able to latch onto the tool that they run down the drill pipe on the coring wire rope.
Yeah, and as long as that thing's been down there, a lot of crud could build up down there.
Kyra was like an expecting parent at this point. This was his baby. This was his research. This was his idea.
So they ran the latch down the pipe, thought they engaged the latch, brought it back up, and there was nothing there.
The dogs themselves that engage the latch, they're not touching anything. They haven't engaged anything.
Set it back down and try again. Sure.
They tried it a second time.
Yeah, we'll see if we can get it this time.
All right. There it is right here.
I brought it back up, still wasn't there.
It's worth a couple more tries, but at some point,
Wait, how many tries?
I don't know.
We just pull the pipe up. Chances are we're going to cut that line somewhere. I can't see us dragging the whole thing out without breaking it somewhere, and we'll leave the rest of it in the hole.
Tried a third time.
So this time we'll get real rough. We're either going to shear some pins or get it, one of the two.
It may have come off.
Thought they got some more weight, and it still wasn't there.
Yeah, we got a little rough with the tool trying to latch it, and again, we didn't get it latched, and so at that point we knew that we could beat on it all night, and it wasn't gonna come out with the wire line.
We had to basically abandon operations and just hope that that entire string came out with the entire cork body.
So we went to bed wondering what was going to happen, and would we have any samples? Would we have something? What was it going to look like when it came up?
And this is actually the oldest cork that has ever been recovered in the history of the ocean drilling program.
How soft is it?
It's real soft, and it's black. It is really strong black underneath it.
Yeah.
There's definitely two layers. Outside's oxidized, inside's reduced.
No, I bet we could probably break that off.
Here's this world-renowned scientist smiling from ear to ear. I mean, I think his mustache was actually touching his ears.
My name is Bubba. I've been a tool pusher and coring technician on the JOIDES Resolution for about 26 years. The dangers of pulling the thermistor string with the cork is if you're pulling out and the ship offsets or the current moves it over, you could take a chance of actually dragging out and cutting the thermistor string.
It's gonna be neat.
We got it.
You guys realize how lucky we are to get all that back, right? I was real happy to see it all come out so smoothly.
Same stuff that we saw this morning.
Lighter stuff is fresher.
Oh yeah, you got a nice tape here. Got a rusticle in here.
Yeah, there are little mini rusticles.
Okay. Coming up, Charlie. What we're gonna do now? We can find here to lay the cork out. We'll lay the cork out. It's a cool environment because everyone has a different background. They all have different training. They all have different goals. They all have different ideals. And yet we're all out here working on one unified project.
I'm out here on this expedition as a cork specialist / inorganic geochemist. A lot of what I do is designing the tools that we need to get the samples. So on our way out here, I've been building some of the wellhead osmo samplers that are going to be deployed within the next week or two. I'm just going to slide all the stuff on here.
Well, we still have to drill all this stuff, and we're not going to have time to do it later.
Okay.
My name's Jeff Wheat. I'm from the University of Alaska Fairbanks. In the perfect world, you have many hours and days to plan out exactly what you want and how you want it.
We only have 2 months out here, so the majority of our time is going to be spent building these observatories and installing them with the future in mind. You know, we're trying to put something down on the seafloor that, you know, the next generation of scientists can come back and use. And our basic osmo sampler is a long coil of Teflon tubing that we connect to an osmotic pump.
Okay. And like, this is the size pump that's going to go in it?
That's the size pump.
Powered by osmosis, and that'll last about 5 or 6 years in the bottom of the borehole and give you a record of the chemistry. It is pretty exciting to see it all happening and going into place.
My full name is Beth Noel Orcutt at the Center for Geomicrobiology at Aarhus University. When we collect rocks from drilling, they're often contaminated, and we have very little control over, how the material comes back. And so one way to get around that is to put material down where we know what it looks like. The colonization experiments that we're using in this observatory, which we call FLOX, an acronym for Flow-Through Osmo Colonization System. So the FLOX experiment allows us to test out different types of minerals and test different hypotheses about which microbial groups like to eat different types of rocks.
We take ground-up mineral material or glass wool, and we put them in cassettes and allow fluid to flow through them.
We're also taking little polished rock chips and loading them onto plastic grids. And those will kind of sit on the outside of the FLOX to allow microbes to attach to the rocks.
And those produce a nice, flat, easy to image surface that we can look at what kinds of microbes are colonizing these substrates.
And then close everything up attach it to tubing, and then they'll get loaded onto the osmo samplers. We use the osmo samplers to pull water through our rocks to explore how the microbial community changes as it goes past these different substrates.
The final fitting of everything into place requires a discussion right there on the cork. And I was seeing these specific corks for the first time. You know, with all the future research that's riding on this, we want to make sure that every detail is covered, everything is double and triple checked.
More than a year ago, we had to have all the pieces fabricated and shipped down here several months ago. So the actual start was 2 years ago, getting all the pieces together. When you then come out here to see and start putting some of the components together, and that's a nice relief. And it's sort of the next stage.
When people first started doing corked observatories, what they would typically do is to provide casing down through sediments to keep them from collapsing. And then they'd leave an open hole where they'd place experimental systems. So it's just basically a great big tube that has your experimental systems within it.
To make the whole cork package work, it's the fiberglass, it's the packers, it's the design for the scientists. They have to have where they want to sample from. And then the whole cork string is designed to get the most out of for science.
For us, in terms of microbial ecology, we're really focusing on in situ technology. So trying to bring the lab to the environment instead of bringing the environment to the lab first.
Being that I'm semi-retired, I gotta admit I'm a little hesitant about coming back out here for 2 months at a time. But I do enjoy this kind of work, of putting these instruments down and getting all this in place. Quarters and eighths first. Okay. And then that one goes the next one up.
The tubing that's used for the casing is stronger when it's being stretched. So by putting a lot of weight at the bottom, you tend to stretch the pipe, and that's where it does the best.
The weight we use are something called drill collars, which are basically heavy wall pipe. In the case of a cork, we're sampling in that lower area, so we tend to perforate or drill holes in the collars.
This allows us to put instruments inside the casing and have water freely circulating and flowing through the formation.
Have they put on the first joint of fiberglass?
Yeah, it's up in there right now.
And then above drill collars, we use fiberglass casing to essentially replace the steel pipe.
We were using fiberglass because of the fact that steel rusts. And a lot of the bacteria that we are interested in studying in this ocean crust are bacteria that use steel for their metabolism, so they can grow off of steel and steel alone. So if you put a big chunk of steel down and then you're trying to study something in situ, you're going to have some problems in interpreting, what's real and what's due to the steel. And so we didn't want to have that kind of influence on our data interpretation.
I had concerns that we have the personnel in the moon pool working, putting it together, and if something broke, well, those umbilicals are going to spool off with it and getting all the personnel out of the way. Not knowing what would happen if there was a bend, would it shatter, would it break and just take off? Nobody's ever run it in open water, and it's the largest string we've ever run, total length.
In addition to sampling fluids from inside this piece of casing, we also sample fluids from the outside through the use of the umbilical.
This was the largest amount of sampling tubes that we've put on a cork before. So you've also got to keep track of what hoses are going to where.
Up above that, there's a packer, basically a big heavy-duty balloon that is inflated with water once it's in the formation, and it seals off different levels of the basement so that when we analyze the fluids from one level, we can make a comparison to the fluids from above.
The wellhead is the last part of the cork, which is the part you see when you come down with the sub or an ROV, the last component of the whole string.
In the case of 395A, it's almost 40 feet long. So they pick that very gingerly up with the crane and lay it in when they bring the pipe in, bring it in, pick it up with a running tool, lower it down through to the moon pool area where we can attach the umbilicals and such. We're going to do a duct test. We'll open all the valves up that need to be open. We'll lower it down. About 10 minutes, we'll purge everything out. Come back up, we'll go down the checklist. We'll close everything up that needs to be closed. Strap on the Osmo sampler, and then off she goes. Make double check them.
All right, that's good. There we go.
Everybody looks good.
Getting the Osmo sampler onto the wellhead was quite an ordeal. You know, it's always an interesting challenge because although it's very heavy and bulky, there's also very sensitive and delicate connections that we have to make.
All right.
So we had no problems at all deploying it. We had no problems with the top latch after that. Everything seemed to be smooth sailing.
We're making a reentry. The whole string only weighed 20,000 pounds, and in this water depth, 20,000 pounds isn't much. So to be able to get the fiberglass string slid over and in the hole, and it went in really nice. I mean, there was no problem there.
Now they should be able to just dunk it.
Almost fair.
Almost 1 meter down. I think it's like 140 before we have any issues to contend with. I've been nervous about the engineering aspects of this project that are new to science since day one. I mean, we knew we were taking a big risk in being the first ones to do this.
We were very close to our mark on the pipe where we should be when we landed out the wellhead. So we assumed at that point that the wellhead was landed.
I think what was happening, we figured out, was those cut packers were hanging up in that hangar. And it was just, they didn't quite want to drop down. Then all of a sudden it just dropped through, and then it came down to our numbers. Because initially we were about 2 meters high. And we picked up, set down, picked up, set down, kept stopping 2 meters high.
It's nice to see everything in there, in the hole.
So we're pressuring up to inflate the packers now.
Pressure up on the packer. Well, he's to make sure he's down and everything.
Drop the pad and then all done.
Yeah. Dropping the pad takes a while, but yeah, pressure up on the packers won't take very long at all. And then we sit there for a while.
So lunchtime tomorrow.
Seeing the landing pad deployed and successfully seated atop the hole is basically the final step. So I wanted to make sure that I was up and I saw with my own eyes that it got in there and it got down and everything was fine. I was not worried at all. At that point, it was a done deal. We had the landing platform, we had the cork in the ground, things were rosy.
The operations manager for SEAM came in and said, looks like we lost our weight. And my first thought is, oh good, we came unjade. And it's like, wait a second, we fought that for an hour and a half, and now mysteriously we come unjade. So I the little hair on the back of my neck were kind of standing up, it's like, that doesn't sound right.
I really had a really sick feeling in the pit of my stomach when we got the camera down there. We all did.
We're just having conversation at lunchtime and kind of casually watching the screen as the camera was lowered after the strings were pulled up.
You can see what we thought was just the J tool, sort of the tool that connects to the cork. We thought it was just that they were recovering.
I think Sam noticed, he's like,
Hey guys, isn't the cork missing from this picture? And they're pulling the camera up at that time, so we got kind of this glimpse of it, and everyone else was like, oh, no, it's there, there's just shadows. And Jeff says,
well, if it was, there would be a problem, and they'd be paging Katrina.
and sure enough, like 1 minute later, Katrina dialed 236. That's when I went upstairs and was told that it had broken off from the top of the plug.
And all the corks that have been deployed, we've never ripped the wellhead off.
As some of you know, we were very close to finishing and succeeding in this hole, and unfortunately, when we went back down with the camera system, we got the video, it's clear that we have a major problem. The cork head is still in the cork running tool, but it has come unattached or broke off from the borehole. Unfortunately, this is the running tool here, and that's the cork head. That's the top part of the bays, and then there's that, and this is the 395A. So we can see that there's some part of the cork. This should be directly right over here. We don't see anything sticking out of there. We can see the at least part of the bottom part of the cork is here. There, Bill, I think that's enough for now. We don't need to see any more. I don't want to see any more. So.
It's a devastating tragedy for the scientists who have been working for a decade to get this cruise, the scientists who secured money over the last 2-3 years to pay for the hardware. It's a tragedy for all of us who have worked for the last year on this. And then we're out on the ship, and the drilling crew, they're applying 20 years, 25 years worth of experience, and they're really proud about what they do. Sorry. Sorry. So they're really proud about what they do. And it's really devastating when it doesn't work out. Sorry, I can't even.
Jerry, Chief Mate, please call the bridge. Jerry, Chief Mate, please call the bridge.
Right when all this tragedy of losing the cork head happened, the captain came to our office and grabbed me and the operations superintendents from our organization and let us know that there was a tropical storm. We had been watching it for a while. It didn't look like it was developing, but as soon as we had this tragedy happen, then we have to spend a day steaming away from our study area.
This is the latest weather forecast tracking Tropical Storm Felipe. Plotted the position for you there. It's moved about 72 miles west-northwest in the last 6 hours. Speed's increased, so we'll have to be well off location by the time it gets there.
I'll notify the engine room. It'll definitely be 2 o'clock to be getting underway. All right, start. We'll switch over to bridge control. Start raising them all.
It's an emotional roller coaster for everyone involved. And, you know, at some point you have to step back and try to maintain perspective. A day or two later, they're rebounding, and now we're back in business. We're doing a new installation. And all that really matters is getting to the end of that situation and having a functioning, scientific subsea floor observatory. Well, the barbecues are something special out here.
It's kind of nice that we can get out in the sun and relax a little bit and have some good food.
This was the first time on this expedition that we'd had that opportunity.
We had some ribs, some chicken, some burgers, some steak, all your fixing of a typical cookout sitting in the middle of the Atlantic Ocean.
Canned drinks, you get the canned drinks. That's the only time you get the canned drinks.
So I think for everybody, it's a just nice break.
Then it was also fun afterwards to see how many people got sunburned with the limited exposure that we've had outside. And
Just take a deep breath before the next hurdle.
Yeah, there's certainly lingering thoughts. Golly, we got to get a good installation in and go forward with that.
Our first operations are to install a new observatory 50 meters west of hole 395A. So then we pulled out of the hole and re-entered with a coring bit for coring basalt.
Recovering core is very difficult. You destroy a lot of things, but in many ways it's kind of the standard type of procedure that's done on these drilling legs. Now this contrasts with really what is the driving force behind what we're doing here at North Pond, which is trying to throw out as many in situ technologies as we can.
Well, the first core is something that all of the scientists get really excited about.
it was fun seeing everybody run to the deck to see the first set of core come on.
The experience of the first core coming on deck was a little bit exhilarating, but also a little chaotic. Everybody wants to see the first one coming up and what it looks like.
Everybody gets excited with the first core on deck. They're very excited. They need to take a quick look at the core, decide how to cut it up, get it in the laboratory, and be able to quickly look and get those microbiology samples out. Each core is really cool, and there could be something really special in it that you've never seen before.
It was the first time I'd ever seen basalt pulled up like that. It was my first core, actual core on the ship that I've seen.
We were really looking forward to see that first core and hoping that it would be massive lava.
Technicians grabbed it, sliced up the core, brought it inside, and we all gathered around gazing on it and seeing what we could see, seeing what was there.
Yeah, and then it comes up and it looked great.
That's pretty good.
Yeah, that's like what?
We drilled only 3.2 meters. That's more than a meter of core.
That's pretty good.
Yeah.
Really amazing, actually.
Yes. Much more than I had hoped for.
There is chemical energy stored in these rocks. If there is energy available, that microorganisms usually find a way to get to that energy.
How is that possible that an organism can survive for 1,000 years without ever growing? That may challenge our paradigms about how life has existed on Earth.
As soon as it starts interacting with seawater, it will change color, it will change texture.
Depending on what we've got and how it broke apart, then we'll just make decisions about, this sample will be for DNA, and this sample will be for doing microscopy, and this sample will be for trying to extract RNA.
In our case, what we're looking for is a process after formation where the microbes have gone in and altered that rock. Veins within the basalt, some cracks, some crevices that we feel that microbes can go in and make a living.
The rock smasher person will sterilize the rock smashing box. They'll spray it down with ethanol and light it on fire, and that's always exciting to watch.
We split the rock with hammer and chisel and a little bit of elbow grease and brute force, which was a blast. That was fun. For me, right now, I'm grabbing the heterotrophs. These are microorganisms that use organic carbon. And so what we want to see is what microbes are mineralizing that organic matter back to CO2. That these microbes breathe their environment just like we breathe our environment.
Up in the lab, we have a core scanner that allows us to get a 330-degree view of the outside of those cores. And so we were surprised because the lava flow turned out to be of a different type, massive lava, not this rubbly pillowed lava. When we look into these rocks, we start the process by slicing them up as thin as the diameter of a hair. We put it in instruments that allow us to measure with point-size precision the chemical composition of different minerals that have formed in different places. And we can determine the oxidation state of metals in that mineral. We can compare that with the state of the rock surrounding the newly formed minerals. And when we do that, we get a glimpse of the action that took place in the transformation. If you have a set of observations like that, and you know what is the global distribution, then you can start to make a reliable assessment of the importance of these processes on a global scale.
So depending on what the scientific objectives are of a particular expedition, so once we've got a nice empty hole, we look at basically how the borehole looks, and we measure that with a caliper just to get a sense that the borehole is not going to, collapse on us. The reason why it's really important is because we don't normally, unless we're very, very lucky, get 100% of recovery on the cores that we're getting out of the hole. And it's the one opportunity to get a continuous in situ measurement as how things actually are beneath the seafloor. We have a fluorescence tool, a unique, brand new tool that we're running specially for this expedition.
The WT instrument, which will allow us to analyze microbial biomass without disturbing significantly or destroying the material that's resident there.
Essentially what WT is a biosensor. It's designed to detect microbes on surfaces. It's got a laser, it fires a laser at a surface, that causes the microbes on the surface to fluoresce, and then we detect that fluorescence. It's the first instrument of its kind. No one's ever tried this before. My name is Everett Salas. I work for NASA JPL. The technology was developed with NASA as a means of trying to find signs of life on other planets. Europa is one of the moons of Jupiter, and there's some ideas that perhaps one day we might be able to core into Europa, say. If we made an instrument that could go into the ocean crust and it's able to detect anything, then maybe we can use that information, use that knowledge. What we're trying to do is just assess what the biomass is like in its native state. So what we've got here is the data from the logging run we did at 395A. And what jumps out is that you can see that there's actually a structure here, and it starts to get larger and larger as you go through this gradient. There's a lot of organic material down here, for sure, all the way down. The expectation was that it would be less than what you see at the bottom of the ocean, but we're seeing more. And the signals are pretty intense, so that people study these cycles because they impact climate, they impact our ability to sustain ourselves. So if now all of a sudden you've got this reservoir of biology down there and it's doing something, then it's going to have an impact that maybe people didn't think about.
After some sleuthing, we went looking for some evidences of what actually went wrong with hole 395A. So when they tried to jam it down, it has these fins around the edges, and those were all crushed in. The throat of the cork was apparently smaller than the diameter of the cork body.
Now you gotta think that that original reentry cone was set on some 25, 30 years ago.
We think that what happened was that some of the data regarding this hole was probably transcribed improperly. We had everything designed exactly properly according to standards. It really should have worked. That should not have been a problem. What happened should not have happened. We really were doomed and we had no idea.
We had trouble with 395A cork in that it bent in the cork body, which I think was a secondary failure. So just to avoid that in the future, we really juggled the plan around for this one. The thing good about having it out here, still get the signs where they're somewhat readable. Still got some clearance. More structurally sound is that.
I'll go with the structurally sound.
Gives a little more clearance.
I mean, part of the reason that thing, the last one, failed was that bottom part didn't get in. And so, is it necessary, I guess is the question.
I can't see any harm except, we're losing these hooks.
The original corks, this part was thinner too. So, they're not going to break through that.
Well, we'll be welding here. We'll remove these two, pull them over to the side and cover them, and then they'll weld it.
You can tell Bubba, he'll show him, get the welder out here. We'll weld one, tack one on, little tacks just to make sure, double check, and then he'll weld them up the same after that. We basically took 2x2 structural tubing, which we had brought out for another purpose, ended up not needing it for that, so we kind of reused it on the corks, and we welded a stiffener on each of the bays.
Getting back to work is one thing, but I still see that cliff at the end of the road with no bridge. I'm hoping that bridge gets built by the time we get there and we get this work in. But the real challenge still lies ahead.
I was incredibly nervous, so nervous like a cat, especially after the troubles that we had at 395A. I really wanted to see a successful hole.
Yes. Get loose. And that's the excitement for the evening. Okay, that's the way it's supposed to work. That was nice.
It was really nice.
Hump Day is always a good day.
It was a two-part celebration. Number one is just that you're halfway through the cruise.
It had a sort of a party atmosphere, knowing that you're on the downside of getting back home.
It also is, of course, our first big celebration for getting the first cork in.
We had some success in moving forward, and at the time, everything was looking good.
It was a job well done for everybody, and everybody was pretty relieved and happy to celebrate that event.
One of the nights when we were doing the hard rock basalt sampling, doing our normal routine, splitting the rocks, dividing them up for different groups. Yeah. Beth and I had the rock, had this piece of basalt in the core box, and when we split it, we split open, and when we both looked at it, we saw this white, milky substance inside the piece of basalt. It struck us as being very different than what we had seen before. This is the first time some things like this have ever happened. And so what we were able to do then, we were able to divide it up. We got a little bit for RNA, a little bit for DNA, and then other was handed to Everett.
WPT is a scanning system that's designed to look at small samples at a high resolution. You take a little rock and then you can make a map of all the organics on that sample. This is the first time that we've actually deployed the instrument onto a ship to look at freshly cored material. So they left me the sample, I took a look at it, and it was pretty lit up. It was pretty fluorescent with respect to a biosignature. You've got a fair amount of organic material on this rock, and it's distributed not homogenously. I mean, it's all over, but it's in specific locations. It's
heavier in certain areas, more concentrated.
Areas where it's more concentrated through these regions in here.
And does it compare this to what you saw in the borehole wall when you scanned the wall with WT?
Well, so the borehole wall information for this hole is mostly organics. There's very little in the way of a microbial signature.
The hydrocarbons that you are detecting on this, compare that to, say, the drilling fluid,
Yeah, so the
pipe
drilling
material,
that stuff gives you a pretty unique signature which fluoresces in a different pattern. And I was looking for that as well, and I don't see that anywhere on these samples.
The thing that was when we cracked this open, Beth and I saw it immediately, but you could easily see through here this white, milky material that was in there.
Right, so there's definitely microbes through here.
That's impressive, and that's something that gives us a little more credibility about what we were saying before, that it was microbial when we cracked it open and got excited about it.
Right.
I mean, that's what we need now, and then I can go back to my samples and see if I can't get something to culture out.
Yeah.
If I can pull that out and see who this guy is, and that'd be fun to link it together and see what happens with it. So,
sweet.
Cool. So what I do in terms of the molecular work that I target is I target RNA. DNA for a cell is like the hard drive in your computer. The RNA is more like the programs you're actively running. So now we can tell, is the cell active? Yes or no? If the cell is active, what process is it doing? What we want to be able to do is to look at and characterize that RNA, because now what we can say is, all right, not only is it down there and buried and locked away and trapped for a very long time, but it's actually active as well. If that's real, then that's awesome. That's huge. That's why we need to keep going, and we need to keep putting these holes in different areas to better understand the Earth's processes as to what is actually happening on our planet.
What we're going to do is set 550, 600 meters cork fairly deep in the basement. That's not going to be a gimme. Although the drilling in North Pond has usually been a decent environment to drill in, but there's no guarantee up here on the slope at this new site that that's going to be the case.
Our objective now is to accomplish that, and that is probably the largest, highest priority, major goal of this expedition.
Yeah, everything was looking good there. We got the renter cone and first string of casing jetted in.
But as with all expeditions, things go every which direction.
We noticed that the torque was getting a little ragged, more so than just the drilling of the rock.
For about 5 hours, we couldn't advance the hole.
We didn't know what was going on, if we were just in a really hard formation or what the problem was.
When we got the bit back up on board, there were a number of us who were very on edge waiting to see what came up at the bottom of the drill string. But our worst fears were confirmed when we saw the bit.
Well, we lost 2 arms off of a 14 3/4 inch bit, and that's just highly unusual.
Everybody was looking at the bit, trying to decide what was in the hole, left in the hole, and try to decide what we were going to do.
It's going to take us 12 hours to build the reentry funnel.
Let's get it going.
Trying to fish that much metal out of a hole is, especially in this deep of water, is pretty much a losing proposition. With the time left, the decision was made to just abandon where we are right here in this hole, start another hole with the hopes of getting it deep.
The hole isn't totally useless. We can actually use it in the future. So what we wanted to do was land a platform on top of the reentry cone so that if we come here with a remotely operated vehicle or a manned submersible, we have somewhere to sit down. So that's what that platform is for.
We're on the right track. We just have to regroup, settle in, and drill this new hole.
And they also say all good things come to those who wait.
Well, we will have to wait longer now.
It's important for us to get deep so that we can get to the sort of the impermeable layer. At 395, it was around 460 meters below the seafloor. So we know we need to get at least that depth at this new site.
All this time that we're putting into this hole, we've got to be really careful this trip. In an instant, you can get stuck. Like I said, we've really got to be on our toes and watch our hole conditions. There's more pressure on everybody in the drilling end.
The risk is if you stop the hole too soon, you don't get the science observatory that you want and the different levels of the oceanic crust as best as you like. The risk on the other side is that you go too far and you have a failure with a bit like we had before, you end up losing the whole hole.
The question is, where do we go from here?
Well, when they talk about this risk, the risk is not minimal, I think. The sooner we start cleaning the hole up and getting out, the less chance we have of getting stuck. If we get stuck now, the hole's gone.
Where is the primary driving force to stop the hole? I don't get it. I would say that right now we should continue on.
More than likely, we would be okay going a little bit deeper.
Keep corn. I haven't seen enough to worry me. The threat's always there, like Mike says, but I haven't seen enough that would worry me. I would core until you want to stop.
I'm a lot more skeptical. You know, I always look at all the things that can go wrong.
The bottom line is things were going well and there wasn't a solid reason to stop.
Everything went well on the assembly and on the running.
The cork head is the very last part that goes on the top of the entire observatory string.
We doubled and triple checked, and then we're strapping on an Osmo pump.
You have one part of your mind that's just sitting there waiting and hoping that it would succeed, while the other part of your mind realizes that, you know, it could still not succeed.
Hasta la vista. See you later, Cork. I don't want to see you back for a long while. I was just wanting to see it get in the water and not come back.
So the way it's going to work is we've got the Osmo samplers laid all over the place here. For everyone else, we're just taking these out to the rig floor, and we're going to go out bottom first outside so that the drillers are going to take it from the bottom end and move it around to the wellhead. And Sam will be on the rig floor helping stuff go down the hole, make sure things go the right side up, and so on and so forth.
That's what I was thinking when you guys.
know, it's not until that very last moment when you unlatch the cork running tool and you leave the site that you know if you've successfully installed it. And you know, each step of the way you feel like you're getting a little bit closer, but the reality is the risks don't change. The risk is there till the very last minute, and so until that happens, you just can't totally relax.
We were worried we would have a repeat occurrence with this nagging issue of the top plug. And this is important because it's ultimately the last final seal of the cork, and you want to have that fully engaged if you possibly can.
Rotating, that's not good.
I was expecting any minute when I saw it apparently rotating, it to snap and break off and it to be returned to the surface, which is just not, of course, what I wanted.
They spun it quite a bit. They did a full rotation on that thing. Definitely not coming back now.
No.
It did look like the cork was rotating, didn't it?
Yeah.
Definitely did.
I know, it looked like it did before.
No, that was the camera swinging.
We'll have to look.
Okay.
Hopefully you're right.
Okay.
Yeah.
Cool.
Oh, it's fine. Yeah.
Cool.
So it wasn't really until the whole thing was over that I was able to take a big sigh of relief and say, yes, we've done it. It's safe in the ground and we can see it next year. At this stage, you know, we know we're going home. We have a lot of work yet in front of us in terms of taking care of business, getting the reports written, getting all our ducks in a line.
It's relaxing to know that the major objective has been achieved.
In terms of getting corks in the ground, there's no doubt that this has been successful. There's still a lot of questions that are out there.
In April, we're going to come back for the next phase of this project.
Basically, we'll be coming back and sampling fluids, also exchanging out experiments on the wellhead themselves.
I'm really looking forward to that anticipation of coming up to the corks and taking our samples off to see if they worked. That'll really be the payoff.
We're just taking part of the next phase of what's been an ongoing experiment here at North Pond. It's really that stage that will ultimately determine the overall success of this experiment.
This is the first part after the drilling leg. We got on the Miriam in the Bahamas, and it was nice and warm and tropical. And now we're heading to St. John's, Newfoundland, where it's anything but tropical. So we go from the nice tropics to the Arctic. What a cruise. Well, this ship is much smaller, very modern compared to even US standards. All the cranes are operated remotely, which saves manpower. We're heading out to North Pond, where we drilled those holes. 2 of them, we're gonna add samplers and collect fluids from the different horizons. And then we're gonna also go over to 395A. That's the one where the wellhead got ripped off. The one junked hole where we were drilling into basement, lost the roller cone and had to stop drilling in that hole. We have some science gear that we're bringing out to put in a smaller version of a cork. And this will be the first cork deployed with an ROV. One of the reasons why we're out here with Jason is because we're going to be down at 4,500 meters. For us to get down there and actually turn valves, make connections.
Scott Hansen, and I am a ROV pilot, technician with Jason. A lot of first discoveries, a lot of new species with Jason. So Jason is the ROV, Medea is the depressor weight. So what happens is, as the ship is riding the surface and it's going up and down, the cable that goes down to Medea is also going up and down. And then we have a neutrally buoyant tether going from Medea to Jason so that we can stay rock solid, pinpoint accuracy while the ship is bouncing around in 10-foot, 15-foot seas. I love my job. I get to drive a robot on the ocean floor for scientific research. How cool is that, right? It is the ultimate video game, and you don't get to press reset. I feel like I'm inside Jason, as if it's a manned submersible, so I am connected. If you all of a sudden lose that, I just have a momentary lapse of disconnectedness, that's when things go wrong. When I'm piloting Jason, one of the main things that I'm focusing on actually is everything. When you can take in 20 monitors, build the 3D image of 3 bodies, the ship, Medea, and Jason, and have it all, the clear picture in your head of what you're doing and where you are.
So we had to recover these fast flow osmo samplers that were deployed on the drill ship.
Yeah, I think that's just parallax.
Yeah.
Because when I'm over here, it looks the same. Here's why. That's right.
Yeah. Wow, we can see it from this close.
Gotta get closer.
So then they cut off the fast flow osmo sampler and placed it on that elevator.
An elevator is essentially just a little float with a weight. It goes down to the seafloor. You can put instruments on it, release the weight, and then the floats will carry it to the surface where you can, bring it on board the ship.
The more broken down it is, the easier it is.
Yep. A few short months ago, I remember shoving my arm in a steam canister. Yeah.
This is PFA. Seawater for.
And that's the end of 306?
That's the end of 306.
When I opened it and started letting the water out, very little water came out. And it seemed highly likely that it never even sampled water.
Basically, you have the fresh water separated from the salt water with the membrane in between, and apparently one of these membranes failed, and so the salt from the salt side came down into the distilled water side, and because of the density, just kept going down through the coil into the samples. All is not lost, but just didn't go as planned.
So for the second experiment that got pulled off of the other wellhead,
And once it came up on deck, started taking it apart.
Oh, good, it didn't leak. Water started pouring out, and we actually collected quite a bit of water.
there we can see a better indication that the pumping system worked in the way that we expected it to.
There's good confidence that we actually pulled water from the formation and were able to subsample it with flocks and osmosamplers.
So we can just do a chamber or one before whatever. Sphalerite, pyrrhotite, pyrite, chalcopyrite, pyrrolite, siderite, glass beads. Okay.
So it looks like they did pump on this one.
Yeah.
In the membrane, you can tell it's all discolored, and you can see the 5 circles on it from where the mylar was, so cool. It was pumping.
Okay.
So the last fast flow osmosampler that I deployed was specifically designed to be deployed on the cork itself from the drill ship. So we were pretty limited as far as how big it could be and its shape and size. This time we weren't limited by the size or anything like that. And also we needed to have something that could be deployed for possibly 2 years. They fit onto this fiberglass platform that I built with a steel rod to be able to lift it up and basically sink it down onto the bottom of the ocean And then the ROV could come and pick it up, move it next to the cork, and then it had to make a pretty long umbilical to be able to reach and plug it in up there. We came up with this idea of collecting the chemicals in situ. We decided to take all that water and pass it through these columns that are filled with different resins, different materials that bind chemicals to it. The advantage of having a 30-gallon drum is you're able to take these very low concentrations of things like radium and lead-210 and concentrate them into a little bottle that then in 2 years will come back and this will be all concentrated and waiting for us right there. Sort of the idea was to put one of the corks and then one at the other site and try and get an age progression of how old this water is in the formation.
It was sad and yet happy to come back and see 395A. Sad because, of course, it's not the experiment we laid out to do in the first place. But it was gratifying on some levels because we could see that the hull was in pretty good shape. We knew that we had skewed the landing platform significantly. We couldn't tell exactly how far, but we knew that it was not on center anymore and it was going to preclude our planned operations into the future.
I was able to actually move it just a little bit, like enough to make us think, oh, okay, all we have to do is make it light somehow. So, in other words, drop down some floats, attach some floats to it, have the floats take most of the weight, and then Jason could actually move the whole thing around.
So I think the plan should be to put the hook right here, so we provide the flotation on that side, and then we come over. Once we have the flotation all attached, we just come over and we grab it. So we get a little bit of an advantage on this side.
Yeah.
We're not trying to completely tip it
Then
over.
when you lift up, the whole thing should just float up because you're just picking up that extra 100 pounds.
And we only have one chance of a connection. I mean, that's, once we connect it, we are unable to release it and connect it again. So we got a plan.
We came up with a solution of several 100 pounds of flotation. So take the hook off of the floats, go over to the grate, and hook it onto the grate. But then it was too much float. It was decided that we don't actually need the grill there. We can sit on the edge of the cone just fine and do whatever work we need in the cone. We were able to just pick it up, drive it out of the way, and release the floats. I guess hindsight, it's even better that it's gone and out of the way.
Rock sampling with Jason is always fun, but it's not always easy to know what you're sampling. These manganese oxide crusts slowly form, so pretty much all the hard rocks that you see on the surface look the same. It's mostly guesswork, so it's always exciting when Jason comes up with the rock samples and you actually take them out of the basket and get to look at them. This is something we are trying to learn is what are the different rock types? What's the structure of the crust around North Pond? How typical is it?
I have no idea what it is.
We actually learned quite a bit about the geologic setting of North Pond. We sampled basalts from different volcanic centers. In the south, there's another high that turned out to be not volcanic in nature. Instead, it is mantle rock. But then this is basalt. It doesn't look to be a breccia because you have mantle material included in basalt here. We knew from the drill core observations that these types of rocks do outcrop in the area. We're trying to figure out what it is.
Grob crystalline, large crystals.
At these low temperatures, the reactions between seawater and rock are very sluggish.
It's like carbonate, yeah, and especially right here on this piece.
One consequence of that reaction is that oxygen in seawater is depleted. The energy source may be something different. It may be organics, but it could also be energy that is released from the rocks in these weathering reactions. On a planetary scale, it's an important process. We just don't know the consequences for chemical exchange and biological activity.
So you should be able to lift this up, put that underneath it.
Yeah.
It's going over the side. So I need one more shackle, so then we'll roll around. We're good.
So the Cork light was a concept that we came up with out on the drilling ship after we had to abandon that hole, the B hole, due to losing a bit in it. This is the first time a Cork has been installed by an ROV, so that's fairly significant historically speaking.
Is there any place I can hold on to here?
You can hold on to those hooks.
Like that?
Yep. How heavy is it? We first figured out how much it did weigh in water to make sure that once we released the boot, it wouldn't float away. And then release the boot, cut away the other dive weights to make it 90 pounds negative in water.
Stay high, get over the cork as best we can tell with nav, and then slowly lower down to the point where we can see it.
We picked it up, moved it over to the wellhead, and placed it first try right through the grate into the well. And so the concept behind it is just to simply seal off the hole and put down some downhole instruments without any fluid sampling program associated with it, just so we can do some experiments downhole over the next few years. When we went to install the whole instrument string inside of it, we actually had some pretty hair-raising moments in terms of when you have the instrument string go down and then you have this tether to the sinker bar and the whole thing kind of collapsed to the seafloor. And then we had to resurrect that and get it inserted inside the hole. So that was fairly exciting, but it all worked out in the end. And that's in part due to the, extreme competence and skill level of the ROV pilots, that they made it. They took basically what we had, which was a pretty bad situation when we had all these parts that collapsed to the seafloor, and resurrected that and managed to get everything down the hole as we planned. Nicely done.
Yeah.
That's nice and solid, huh? Good. All right, so I think we're done here. We're done with this part of it.
I found it to be enormously gratifying to be able to resurrect the bee hole and bring it back to life, and to install successfully an instrument string within that. Things don't always go as you plan them to go, and you come out to sea. When you can take a problem and make it into a promise, that's really a great thing.
By using the ROV to install a cork, it opens up a huge range of possibilities.
So essentially, the ocean drilling program, any place that they've drilled into the oceanic crust, they can go back to it. So there's something like 55 of these holes around the globe. And I think the Cork Light is like the perfect experiment that we want to, take out to these holes.
Part of the excitement of this whole North Pond project is that we're trying to go after a set of questions, a set of things that we're interested in that no one has really looked at before, right? We're trying to study life in deep oceanic crust, which is a huge potential reservoir for life, and we know nothing about it.
At this stage, we're going home. Early indications are that everything has been fairly successful, and a lot of the data comes from the pressure measurements that we've been making out here. Those are giving us our best indication as to how things are going. Basically, what they tell us is that the boreholes are isolated from the seafloor. There's also some early indications that the different intervals as a function of depth are isolated from one another. There's small but measurable pressure differentials between them. And so those are telling us that things are set up as we intended.
With all these questions and doubts and having the first wellhead get ripped off, everything came out smelling like a rose. What we set out to do 6 years ago, we've completed. And one of the things that keeps me coming back doing all this stuff is that the people are great. Everyone from the Bubbas to the cooks to the other scientists, everyone is out here enjoying what they do, which is great. I mean, you know, there's a lot of jobs out there where people hate going to work every day. I like going to work.
I take a lot of satisfaction from what we've accomplished so far. The fact that we've set it all up properly, I think, is a huge accomplishment.
We are just getting started with this line of research. The studies at North Pond, I mean, chapter 1, I would say, is done, but there's many more things we need to do.